Hi there! Did you know that about 1 in 12 people in the world suffer from diabetes? Out of these, 10% are type 1 diabetics, meaning the insulin-producing cells in their pancreas, the beta cells, are defective. Could we culture and transplant into these people beta cells able to replace the diseased ones? First, we need to understand how cells arise in the body. When sperm and egg fuse, they give rise to the zygote, which in turn divides multiple times to form the embryo. Cells in the early embryo have the potential to originate any of the more than 200 different cell types found in the human body. To this characteristic we call pluripotency. Cells then specialize, acquire functional maturity and lose potential during a process termed differentiation. Why are cells in the same organism so distinct, even though all share the same genes? A complex mix of internal and external clues determines cell fate, activating and deactivating certain genes. In highly differentiated cells, parts of the genome are permanently blocked. Waddington illustrated this concept in his landscape. Just as marbles can travel uphill after falling from the top of a mountain, mature cells don't usually revert their specialization. But could they? Shinya Yamanaka decided to tackle this question and try to obtain stem cells from specialized cells. To achieve these, he first inserted a set of 24 genes associated with pluripotency into mice fibroblasts. They started to change shape and became very similar to embryonic stem cells. Then, Yamanaka repeated the experiment, reducing the number of genes and trying different combinations. His conclusions were surprising. 4 of the genes were enough to reverse fibroblasts' specialization. The cells obtained were called induced pluripotent stem cells. These could later be induced to differentiate into other cell types, such as nerve or muscle cells. In 2012, just 6 years after publishing his results, Yamanaka received a Nobel Prize. His discovery has endless applications in medicine. iPS cells can be used to culture defective cells in vitro, providing a tool to study disease development and drug effectiveness. They may replace embryonic stem cells in research and therapy, thus overcoming various ethical issues. And they could help type 1 diabetes patients by providing them with new healthy beta cells. It's an open field of research and there's a lot to discover. Will you join?